kudos for you. So in the lab, we are very excited about microRNAs. I'm going to give you a couple of slides that hopefully will get you excited too. And so the first one shows you the relationship between, okay, let me get a pointer here. Okay. Between biological complexity as increasing number of different cell types and the number of protein coding genes. Each square is a different organism. So you see bacteria here and humans all the way up here. And you might notice that there is a problem, right? From the dawn of multicellularity with this amoeba here all the way to humans, it doesn't really seem to be an increase in the number of protein coding genes. And so where is the genetic information that we need to achieve this massive increase in complexity? And the answer is, well, probably here. So if for the same organisms, you now plot the number of non-coding genes, what you see is a beautiful linear relationship. And what this suggests is that evolution did not need to add more building blocks, but instead to add mechanisms that control gene dosage in space and time. And this expansion of the non-coding RNA repertoire is particularly evident if you look at our favorite class of non-coding RNAs, that is microRNAs. And this graph shows you the new microRNA families that emerged in the last 400 million years. And you can see that compared to simpler organisms in mammals and primates in particular, you really see an explosion in the number of new microRNA families. A lot of these families are enriching the brain, and this suggests that microRNAs are necessary to assemble and maintain the mammalian brain. But what are microRNAs? In their mature form, you see this squiggly red line here. They are loaded in this Argonaute complex, or AGO, and they recognize their messenger RNA targets based on sequence complementarity to a microRNA recognition element, or MRE. Once this interaction occurs, it recruits a family of protein called TNRC6. These are scaffold proteins. I'm not going to go through details, but what you need to know that through TNRC6, it mediates the repression effect that microRNAs have on gene expression. Of course, there are hundreds of microRNAs in each cell type that we know of, and each microRNA targets hundreds of messenger RNAs to form a microRNA target network. And I really want you, sort of from now on, to start thinking about microRNAs as a network with their targets and not as single entities. So are these networks important for the development of the brain? Indeed, they are. If you remove this enzyme called dicer, that converts an inactive pre-microRNA into the mature form of the microRNA that I just illustrated before. If you remove it from cortical progenitors, you can see here at P0, this is a normal mouse brain that we're looking from the top. And this is when we remove dicer, the cortex is pretty much gone. We can slice it up, cortex is gone, slice it up, hippocampus is gone. So very dramatic phenotype if you remove regulation of gene expression by microRNAs. And from this sort of early study that I just showed you, I would say that the most important questions that we need to solve in the field are where and when a microRNA is important, so which cell type and at which stage of the neuronal life are microRNAs important. And something that my lab is particularly interested in is how. So what are the molecular mechanisms that microRNA use to instruct development of neurons? And I'm going to argue that despite this field being now around for almost 20 years, we still know relatively little. And this is because of both technical and conceptual limitations. And in the first 4 years of the life of my lab, we've been pushing both technological development and sort of pushing new ideas to overcome these limitations. So let me start with technical development. So the tool that I showed you before, the paper that I showed you before, dicer knockout in cortical progenitors, uses an EMX1 CRE line that kicks in around embryonic day 9. And as you can see from sort of the major things that are happening in this for these preoperative neurons is just before neurogenesis, and then maybe a week and a half before neurons start differentiating. But you can see from the title itself that what the authors conclude is that microRNAs are not important for the expansion of neuronal progenitors. So are not important for this process, but they're important for this one, okay? And so the I would argue that here there could be two hypotheses to explain this data. One is what the authors say, that there is a stage-specific role for microRNAs that are important for differentiation, but not for neurogenesis, or that there is a delay in the onset of the phenotype. Even worse, if you look at a different cell type of the brain, this is Purkinje cells of the cerebellum. This Cre line, A7 Cre, kicks in around P4, so postnatal day 4, but these cells have a very prolonged differentiation in postnatal life. However, when you do dicer knockout with this A7 Cre line, the authors observed absolutely no effect on differentiation, and you need to wait 8 to 10 weeks to be able to see anything going on. In this case, is degeneration of Purkinje cells. And so there are two possible conclusions again here. One is that maybe microRNAs are cell type specific, so they might be dispensable for this specific cell type, or that there is a very long delay in the onset of the phenotype. You might have already realized that that's sort of the hypothesis that I'm pushing in both cases, with data in the lab showing that this is indeed the case. And what's happening, the reason why you have such a big delay in the onset of the phenotype is because microRNA half-life, particularly in post-mitotic cells like neurons, can be very long. So the microRNA is sort of shielded, both at the 5' and 3' from the argonal complex, and so the RNases cannot really degrade it, and so abundant microRNA can survive inside cells that lack dicer for weeks and weeks. Okay, so everything we've done so far in the field using dicer knockout is probably not very good if you're looking at development of the rodent brain, in which things happen very rapidly. So we need a much faster tool to be able to block microRNA function. And so what we did is we took advantage of this peptide that has been developed by Gunter Meister's lab. It's called T6B, this peptide, and is the portion of T966 proteins that has high affinity for argonauts proteins. And this peptide was designed to be able to map microRNA-Target interactions, basically by IPing argonaut microRNAs and RNAs. What we thought is, well, maybe if we express enough of this guy, we might be able to have some sort of dominant negative effect and prevent T966 protein from binding to argonaut, and so therefore inducing de-repression of all microRNA targets. Does it work? All right. Pay attention here, because this particular tool will come back a couple of times during this presentation. And this is what we call the microRNA sensor. Is a virus that has two promoters. One promoter is driving BFP, that is independent of microRNA regulation, and a second promoter is driving mCherry, that instead is repressed by a very abundant microRNA 124, very abundant in neurons in particular. So what we normally get is that you have these two neurons that are infected with this construct, have high level of BFP, and you can see a very low level of mCherry. Then we use a second virus, in which what we're doing is we are expressing that peptide that I told you that blocks microRNA function and is fused to YFP so that we can see it. And you can see in the bottom here, these two neurons that are expressing the sensor are also expressing T6B, so microRNAs are blocked, and this is why you see a nice rescue of mCherry level. This YFP that you see here is our control, and the control is basically we mutated the amino acids that are important for binding to AGO, so this is just a YFP that is floating in the cell and is not blocking microRNAs. So if we quantify this effect, you can see here, this is when mCherry is repressed, and this is a de-repression that is induced by blocking all microRNAs. And this sort of de-repression is not too far from our internal control. That is when we mutate all the bases, many bases that are important for the binding of 124 to this MRE. So basically 124 is not regulating mCherry anymore. Again, mCherry becomes independent from microRNA regulation. We're not too far. Okay, does this peptide has any biological effect? Indeed it has. So if we infect cortical neurons in culture, you see this is a normal cortical neuron with beautiful decarbonization. And if we block microRNAs, you see a very dramatic decrease in the complexity of that dendritic arbor. So we're very excited about this tool because this is a peptide and it's virally delivered. So we can do some virus engineering, some protein engineering, and make it cell type specific, make it inducible, reversible, restricted to different compartments of the cell. Basically, we can achieve the highest spatial and temporal resolution in blocking microRNAs that we've ever had. And so, we're very excited about this. I'm going to give you a couple of examples of what we're doing. So first of all, as I was telling you before, probably the lowest hanging fruit for the use of this tool is the differentiation of Purkinje cells. I'm going to remind you, the paper that is out there shows that if you use a 7 credit dicer knockouts, so remove dicer from exclusively Purkinje cells, you get absolutely no phenotype. So this is a normal maturation of these cells, and at the end, P21, they are exactly the same using this strategy. So what we did is we cloned an L7 promoter before our viral TCSP YFP. We injected it in the ventricle at P0, and it starts being expressed around P4. And I can show you the rest of the brain. There is absolutely no other cell type that is labeled by this strategy. And you can see very nice labeling of Purkinje cells that are developing across time. And so what happens if we block microRNAs? Well, the effect is very dramatic. You can see here in normal cerebellum. This is a sagittal slice of the cerebellum. This is what it should look like. And this is when we block microRNAs. You can see by eye, it is a lot smaller. We can quantify it. This entire area of these slices is 30% smaller. Keep in mind that Purkinje cells are less than 10% of the cerebellum. So this is a very dramatic effect. And if we zoom in on individual Purkinje cells, you can see the beautiful arborization of these cells in control condition. When we block microRNAs, that arborization is severely compromised, as you can see from this shoal analysis here. Now, the next question that we ask is, well, are we affecting also behaviors that are linked to Purkinje cell function? And indeed we are, because we're really messing them up. So for example, Purkinje cells are very important for motor coordination. So if you do the rotor test, you can see in black, that's what animals normally learn how to do it pretty quickly. And this is almost completely ablated if you block microRNAs. And recently, Purkinje cells have been linked also to sociability, and in particular to the ability of a mouse to prefer interacting with another mouse instead of to a new mouse instead of a new object. And that preference is definitely diminished when we block microRNAs in Purkinje cells. All right, so one of the things that we got really excited about, sort of, protein engineering with this TCSP is normally, we do dicer knockout, microRNAs are gone and is irreversible. Eventually, microRNAs, lack of microRNAs kills the cells, so it's not a particularly good strategy if instead what you're looking for is maybe a phenotype that has to happen in the adult or late during development. So what we developed is a version of this TCSP in which we fused a diagonal moiety that has a name called DHFR, and this protein is produced and is very rapidly degraded, so it never really accumulates. But if you add TMP, the small molecule TMP, what this does is it binds in this pocket here, stabilizes the diagonal moiety, and allows TCSP to rapidly accumulate and execute this function. So this is just to give you a snapshot of what happens in vivo. This is after maybe a week of the RNA being present, but as you can see, there is basically no protein accumulating. And then 24 hours after one single injection of TMP, you have nice levels of TCSP, and by 96 hours, they're back to normal. So that gives us a nice tight window in which you can block microRNAs and then revert conditions to normal, okay? So this also allows us to start testing by giving multiple injections, one injection a day. Well, what about our weeks, are the 3 weeks that are necessary for the dendritic development of Purkinje cells, are they all the same, or are some more important than others, right? And so there's some preliminary data showing you that both the first and the second week seems to be important, but the second week is more important than the first. And we know this just by looking at the morphology of the cells where they're developing. The second week when most of the dendritic development is happening. So this makes perfect sense. Now, with this tool that we call TCSP, we can really start to address the where and when microRNAs are important. But once we know the microRNA is important for a certain cell type at a specific developmental stage, what we still need to figure out is what the mechanisms are. And to begin to get a mechanism, the first thing that we need to do is map miRNA-Target interactions. And to be able to do this, we use this technology called AGO2 ECLIPSE-seq. CLIP stands for cross-linking and immunoprecipitation of AGO2. So what we have is a conditional version of AGO2 that expresses a tag. So for example, we can express it exclusively in Purkinje cells. And then we use UV light to cross-link this interaction, freeze this interaction between AGO, the microRNA, and the target. And then we use RNases to digest the part of the messenger RNA that is not protected by Argonaut. At this point, now we can then use the tag for IP and then send everything for sequencing. And what we get is this, places where Argonaut was frequently binding. It's going to give us peaks in the transcriptome. And recently, we have added a interesting sort of twist to this technology. We ligate the microRNA in the target, so we get what we call miRNA-Target chimeras that allow us to tell, okay, there is a peak here, but now we also know which microRNA was binding. And doing a lot of this mapping in different cell types, what we now start to understand is the properties of these microRNA-Target networks. And this is a cartoon of these networks in which circles are the microRNAs, squares are the targets. The size of the squares or circles means how abundant these things are, and the lines that are connecting them are telling you the microRNAs are repressing these targets. And you might have noticed right away, so this cartoon is numerically actually pretty accurate for most of the networks that we described. And what we notice is there is two characteristics here, that there is redundancy, and there is the fact that these networks are not homogeneous. So you can see at the center of this network, there is a lot more happening than at the periphery of the network. And the way us and others have been trying to understand sort of mechanisms in this network has been pretty crude and sort of naive, and is to find microRNAs that are highly abundant and do loss of function of individual microRNAs. And this gave us some important results, and it often gives us strong phenotypes. And it makes sense because these abundant microRNAs can constitute up to 20–30% of the entire network. So of course, if you knock it out, you're going to have some sort of a subset of the phenotype that you get by knocking out the entire network, right? But the strategy is particularly cumbersome if then you're trying to get a mechanism, because each of these microRNAs targets hundreds of targets. And when you remove this microRNA from the equation, these targets go up maybe 10–30% in terms of protein. And so your phenotype is really distributed and very hard to study. But what this approach told us is that there are nodes of vulnerability in the microRNA-Target network. And in this case, these nodes are on the microRNA side. And so in the lab, we thought, okay, well, can we find nodes on the target side? And indeed we can, and we call them microRNA hotspots. And these are a small subset of targets that see disproportionate amount of repression by microRNAs. And what we think, sort of the working hypothesis in the lab right now, is that these microRNA-Target networks really evolved to take care of gene expression levels of these hotspots, and that everything else that is happening in the periphery is some sort of playground for evolution. Maybe a million years from now is going to be important for something, but not necessarily right now. And the reason why we think that they have to be repressed so tightly by microRNAs is because misexpression of microRNA hotspots is probably detrimental for the cell, or maybe for development to occur properly. And the other reason why we're excited about sort of this conceptual leap that we've done in the lab is because when you are able to de-repress entirely one of the hotspots from all the microRNAs converging on it, you see a large increase in protein, no longer 10–20%, but more like 5 to 7 folds. And so if you find any phenotype, then this is strictly linked to the lack of repression of a single gene, and this is mechanistically a lot easier to study than the other way around. All right, so that was a cartoon. This is actual real data. This is a miRNA-Target network from developing interneurons at P0 in the cortex, and you can see these are called bipartite networks. At the bottom, you have microRNAs. At the top, you have the targets. This looks like a solid line, but really is something like 5,000 targets that are very close to each other. And the lines are connecting the microRNA and the targets to tell you that those microRNAs are repressing the targets. So how do we find the hotspots? Well, we need to take into account another variable, not just how much microRNA repression these genes have, but also how abundant they are. And this is what we do in this graph. Each of these dots is a different gene. And we basically plot how many reads do we get in the clip for each gene. And then we plot for the same gene how many reads we're getting for a normal RNA sequencing experiment. And the reason why we're doing this is because, of course, when we do an IP, is more likely that you're going to get sort of clip values if the gene is highly expressed compared to if it's lowly expressed. And you see this regression line, blue line, tells you exactly that. The more abundant a gene, the more likely that you're getting clip signal. So we designed this clip score. So basically, the further away you are from this regression line, the more you're looking at genes that, independent of their expression level, are heavily repressed by microRNAs. And so we can really, based on these colors, we can really sort of select a subset of these hotspots that are very, very heavily repressed by microRNAs. And this is numbers just to give you an idea of what kind of numbers we're talking about. And so another sort of exciting use of our TCSP is that, we can very rapidly block microRNAs in vivo. You can see here, this is hippocampus at 0 hours, 3 hours after TMP. You not only have good levels of TCSP, but you also have a nice de-repression of the sensor. And so you have this very tight window in which you can de-repress microRNA targets and hotspots, and then do translatome and proteome, cell type specific translatome and proteome, and really identify and prioritize which are the most repressed hotspots that are happening in the cell type that you're interested in. All right. Once you've identified these hotspots, of course, you need to figure out what they're doing. And so this was another area of pretty intense development in the lab. We needed something that allowed us to sort of at scale test these microRNA-target interactions. And the way we solve this problem is by using a CRISPR, an RNA CRISPR-Cas system. So you're all familiar with Cas9 that recognizes DNA, but there are Cas proteins that like RNA instead of DNA. And so what we do is we use a dead version of this Cas that does not chop the RNA, but we can program it in a way that it will recognize a specific microRNA recognition element and prevent microRNA from binding it. And so does this work? I'm using the sensor again, and you can see here, normal condition, very little red, high levels of blue. And this is our policy control when we completely mutated the mCherry, so there is no regulation of microRNAs. And you can see we have some sort of intermediate phenotype when we design a guide RNA that targets this specific 124 mRNA. And this is quantified here. You can see here, this is like our policy control. So we have 57% of this 100% de-repression. We're working on making this better, but we're pretty happy with this result. And this is the reason why, is because the best tool that is available out there that is called target site blocker only achieves 40% de-repression. It's very expensive, so not scalable, and also is not subtype specific. So we have a better result, and we're also scalable and also subtype specific. And we're not actually that far from the TCSP level when we're blocking all microRNAs. And so this is just to show you, of course, that was an artificial microRNA-target interaction. This is a real microRNA-target interaction. This LFNG microRNA 125 interaction, the clip data showing you a nice peak. And you can show here just by blocking one of these sites, we get a nice de-repression, showed also here. And so we want to apply these strategies to Purkinje cells. As I showed you before, very strong effect with TCSP. And we found both microRNAs that are highly enriched in these cell types. So we even found microRNAs that you can see here, like this microRNA 206, that is almost exclusively expressed in Purkinje cells. If you compare to pyramidal neurons, you see it here is basically expression levels of microRNAs in pyramidal neurons as Purkinje cells, and most of them are expressed at similar level. But 206 is very abundant in Purkinje cells. but not in pyramidal neurons. So we're interested in trying to figure out if this has anything to do with the unique architecture and function that these neurons have compared to other neurons in the brain. And then, of course, we found a bunch of hotspots, and we found exactly which microRNAs are binding to it. And we're developing and we're designing viral ways of delivering both the Cas and these guide RNAs to be able to block these guys. All right. So I hope I convinced you that we have tools to address where and when microRNAs are important, TCSP to fast microRNA loss of function, and then for mechanisms, basically AgoClip-seq plus microRNA spots and RNA CRISPR to really get a mechanism. And this is the people that are doing the job in beautiful San Diego, California. I want to just highlight Fede, Nori, and Jess did most of the job that I presented, and collaborators here and the source of funding. And please ask questions.